page 38 of our notes today. We're actually going to start talking about sexual reproduction. So remember that before we talked about different types of asexual reproduction, and that mitosis is the process that can allow that to happen, where you make identical daughter cells. But in sexual reproduction, this is where we'll actually talk about two sources of genetic material, and that at the very end, they'll actually produce daughter cells that are different from each other. So sexual reproduction, we have our two sources of genetic material, usually one from sperm and one from egg, and two parents are going to contribute half of their DNA. So if you look at the picture that you have on the right-hand side, here's the female, and she's going to produce the egg cell. Here's the male that's producing the sperm cell. Humans have 46 chromosomes. So mom has 46 chromosomes, dad has 46 chromosomes. When they make egg or sperm, the egg or sperm are only going to have 23, half the number of chromosomes. When sperm and egg join together, this process is called fertilization. Okay, now let's put a Z right there, fertilization. And that cell that was sperm and egg actually become our zygote. The zygote will have 46 chromosomes. So it's actually really important that mom and dad only donate half of their genetic information. If they have more or less, that's actually going to affect the organism that is going to develop. Every species has a very specific number of chromosomes, and humans just happen to have 46. But a dog might have 20, a monkey might have 12, okay? It just varies. And so your zygote is actually the first cell that you are, okay? That's when egg and sperm have joined together. The DNA from the sperm actually enters into the egg, not the whole sperm cell. And then it'll actually go through mitosis to form into an embryo, a developing baby, okay? The advantages to sexual reproduction is that there's increased variation, okay? The DNA from the egg and the sperm are very different from each other. They're actually even very different from mom or dad. There's a little mixing that happens before it gets to our zygote. And the disadvantage is that this requires finding a mate, and it takes longer, and it actually takes more energy, and you can also get some undesirable variation sometimes. For some animals, they might get the fur color that's not good for the area that they live in, okay? Or they might not have a thick enough fur coat. Now, with meiosis, there's several differences from mitosis. You still have the same phases as mitosis, so you still end up having PMAT, prophase, metaphase, anaphase, telophase, except that in meiosis, we actually have two divisions. So the cell divides twice, not just the one time, and we're not producing those identical cells at the end. So in meiosis 1, the first time that the cell divides, in this picture they're showing you our parent cell. The parent cell, right here we're just preparing the DNA. Okay? So you can think of this as the parent cell during interphase. Now this is going to begin with a diploid or somatic body cell. Somatic means body cell. What you're going to do is you're going to actually reduce your chromosome number, and you'll actually end up with two haploid or different daughter cells. So here's our parent cell, goes through interphase so the DNA can replicate. So here's our cell that's ready to divide. It will go through PMAT 1, okay? So the first time the cell divides, and now we actually cut the number of chromosomes in half. At the very beginning, we had 4 chromosomes in this example cell. Those 4 chromosomes have doubled, so now you have 4 doubled DBL chromosomes, CHRS. And they go through PMAT, prophase, metaphase, anaphase, telophase, and now each of these daughter cells have only 2 doubled chromosomes. Okay? They're going to go through PMAT again. So here you have PMAT 2, prophase, metaphase, anaphase, telophase 2. And at the very end, each of these 4 cells will have half of the number of chromosomes. They'll actually have 2 single chromosomes. So up here, if you notice, these chromosomes were 4 single chromosomes as well. So you end up with half the number of chromosomes. And this would become your sperm or egg cells. So in meiosis 2, when we go from here's PMAT 1, we have 2 doubled chromosomes, to PMAT 2, the second time it divides. We're reducing the genetic material, so we're cutting that material in half, and by separating those doubled chromosomes. Because remember, in this picture right now, they're attached. When they're doubled, they start to look like those X's. The result is you have 4 genetically different haploid cells. Haploid is going to mean half the number of chromosomes. Okay, where diploid meant the total number of chromosomes. So these cells are now called gametes. Those become sperm or egg. And eventually they can join together. They'll each have 2 single chromosomes. Sperm will have 2 single. Egg from the mom will have 2 single. And they'll join together, share that DNA, and they'll have 4 single chromosomes and be able to become a new organism. So meiosis, one of the benefits of meiosis is it does allow for that genetic variation. The genetic variation, or the differences in that DNA from the parent and from each individual sperm cell or each individual egg cell, comes from one of these 3 processes. So you have crossing over is the first way in which we have genetic variation. How do we have different DNA in each sperm cell or each egg cell? And the chromosomes will actually exchange similar sized pieces and increase variation. So you have chromosomes, 2 chromosomes, a pair of chromosomes from one parent. Those chromosomes will come really, really close to each other, and they'll actually switch some of that genetic information. And once they switch that genetic information, each of these chromosomes are now very different from each other. They have different genes on them. And so each section, or each chromatid here, when it finally separates to become single chromosomes, will be different from each other. And so you'll have your different sperm cell or your different egg cell. With random assortment, this means pairs of chromosomes separate independently. So when they're going through that division, when they're actually going through anaphase and the chromosomes being pulled apart and pulled into different areas of the cell, they actually have no rule to it. It can be this blue side can come over here, this red side can come over here. It doesn't matter, it's random. That increases our genetic variation. And then the last way in which genetic variation is created is through random fertilization. The sperm and egg that join together are just very random. It can be any random sperm cell, any random egg cell. There's no way to really track which egg cell is going to be there and which sperm cell is going to get to the egg first to fertilize the egg. So all 3 of these things will increase the genetic variation of the organism that's created at the end. Now there are some problems that can come up with meiosis, okay? And with meiosis, you're making more of you, more of the whole organism. 3 problems that can occur, first of all, is a mutation. A mutation is any random change in DNA that can happen during DNA replication and can produce a variation that is good or bad. Not all mutations are bad. Some actually can have some benefits, but some mutations are bad. Non-disjunction is another problem that can occur, and this is when the chromosomes don't separate properly. You had that replicated chromosome, but it didn't pull apart fully. And when it doesn't pull apart fully, now you have some cells at the end, some gametes, sperm or egg cells, that either have too many chromosomes, it's only supposed to have 2, this one has 3, and this one has 1. And that can actually cause what's known as aneuploidy. Now, aneuploidy is when you have a fertilized zygote or a fertilized egg, sperm and egg joined together, but now they have the wrong number. Non-disjunction is talking about, okay, this one gamete has the wrong number in it. Aneuploidy is talking about now we have a zygote with the wrong number of chromosomes. And when it has the wrong number of chromosomes, we can get some disorders like Down syndrome. And Down syndrome is also known as trisomy 21. Okay? For your summary box for this section, I want you to pick 2 main differences that meiosis has compared to mitosis. Remember that meiosis is about making your sperm and egg cells. It's not about making a whole new organism yet. They have to go through fertilization first. But meiosis, you're making your gametes, sperm and egg. And how is the sperm cell or the egg cell different from the cell, the daughter cell that's produced in mitosis? So look back in your notes and figure out 2 ways in which they are different.